We are going to study the structure of DNA. The science of molecular genetics began with the determination of the structure of DNA. You are aware of genetic engineering, gene cloning, forensic science, and GMOs. All this was possible only due to the discovery of the DNA structure. This is one of the most dramatic in the history of science and has been the subject of many historical treatments. If you ask a question that, sir, why knowing the structure of DNA is important, then my question to you is that how can we know that in which form DNA has stored information of life, how DNA replicates, and how it transcribes, and how can we know the process of mutations, and as well as if we talk about the genetic engineering, how it was possible. All it became possible after knowing the structure of DNA. That's why to study the structure of DNA in deep, it is very important. Now we will discuss the early clues about DNA structure till its structure discovery. So the Phoebus Levine, a Russian scientist who proposed the tetranucleotide hypothesis, suggesting that DNA structure is made up of equal amounts of four nucleotides that are A, T, G, and C, arranged in a repeating pattern like ATGC, ATGC, ATGC. According to him, this made DNA too simple and regular to carry complex genetic information. Although this idea was later proven incorrect, it was an important early step that guided scientists towards a deeper understanding of DNA structure and then its discovery. After Phoebus Levine, another scientist contributed to this field and he is known as Erwin Chergaff. He formulated a rule that is known as Chergaff's rule, and in this rule he stated that amount of the adenine in DNA will be always equal to the amount of thymine and amount of guanine will be always equal to the amount of cytosine. And he also calculated the ratio of A plus T and G plus C, and he showed that this ratio of G plus C and A plus T differs among species. In this way, he provided key evidence that DNA has specific non-repetitive structure. Erwin Chergaff, by formulating Chergaff's rule, he disproved the tetranucleotide hypothesis. Tetranucleotide hypothesis was suggesting that DNA is made up of four nucleotides which are A, T, G, and C, and he said that this unit will repeat again and again to form long stretch of DNA. But Erwin Chergaff disproved this tetranucleotide hypothesis. He demonstrated that DNA-based composition varies between species, and his findings were crucial for James Watson and Francis Crick in proposing the DNA double helix. Now X-ray diffraction studies of the DNA started, and the first scientist who did the X-ray diffraction studies of DNA, that was Maurice Wilkins. He began early work on the DNA structure using X-ray diffraction, and he proposed that DNA might have a regular ordered structure and also produced initial low-resolution X-ray images of DNA. Maurice Wilkins shared important data including photo 51 with other scientists, but this photo was not the property of Maurice Wilkins. It was a stolen photo by Maurice Wilkins from the Rosalind Franklin property. But this work laid the foundation for further detailed analysis. Rosalind Franklin was also studying the structure of DNA through X-ray diffraction, and she captured famous photo 51 showing the helical structure of the DNA. She captured actually many photos and each photo was numbered. That's why this photo 51 was also from one of them. She also calculated some key measurements like the diameter of the helix. She said that it is almost 2 nanometer and the distance between the stacked bases is 0.34 nanometer, and she also provided strong evidence that DNA is double helix with repeating units, and her precise data was crucial for building the correct model of DNA. She also determined that DNA has two forms, A form and B form, and in this way she contributed in the discovery of the structure of DNA. Stolen data behind the double helix discovery. What is the actual story? Maurice Wilkins and Rosalind Franklin were working in the same university. Maurice Wilkins stole a photo 51, from the Franklin's property and showed it to other scientists. A report containing Franklin's unpublished data was also shared with the Francis Crick through internal communication, and the matter of concern is that she was not told that his data is being used. And Watson and Crick used this data to understand helical structure of DNA, precise dimensions of this molecule, and she was not directly informed that her data is being used. This raised later concerns about scientific ethics and credit. James Watson and Francis Crick also made an attempt to discover the structure of DNA by assuming a 3D model. James Watson was a young biologist. He was interested in genetics and DNA structure. He was inspired by X-ray diffraction studies of DNA and also wanted how genetic information is stored. He was eager to learn. Second was Francis Crick. He was a physicist. He turned to biology and he studied molecular structures using theoretical approaches. And he was also interested in applying physics to biological problems. Watson and Crick used available data to construct a 3D model of the DNA. They applied Chergaff's rule like base pairing rule of adenine will bind with the thymine and guanine will bind with the cytosine. Also interpreted X-ray diffraction images like the photo 51. They also took information from that photo and they built physical models using metal plates like you can see in the picture. Final model was having features like double helix structure, two anti-parallel strands, and complementary base pairing. Watson and Crick showed that DNA is composed of repeating units which are called nucleotides, and each nucleotide is consisting of deoxyribose sugar, then phosphate group, and nitrogenous bases, which may be adenine, thymine, guanine, cytosine. And the nucleotides are linked by phosphodiester bonds, forming a sugar phosphate backbone. Mean in the strands there is sugar, there is phosphate group, but the bases are embedded in between the two strands. Two strands run anti-parallel and coil into a double helix, and base pairing rule was specifically the same as stated by the Irving telegraph that adenine binds thymine and guanine binds cytosine. To completely understand the structure of DNA, we must have to understand first its basic unit, that is nucleotide. In DNA, it is often called deoxynucleotide, and each nucleotide has three components which are phosphate group, deoxyribose sugar, and nitrogenous base. In this figure, you can appreciate this is phosphate group, this is ribose sugar, and this is nitrogenous base. Now we will study the each component of deoxyribonucleotide in detail, and the first we are going to study is deoxyribose sugar. This sugar belongs to a group that is pentose sugars. These are named pentose sugars because they are containing 5 carbons in a ring-like structure. Here you can see the ring of this deoxyribose sugar. In this ring, carbons are present on the corners of the ring, and these carbons are numbered as 1 prime carbon, 2 prime carbon, 3 prime carbon, 4 prime carbon, and 5 prime carbon. This numbering is given as 1 prime, 2 prime to differentiate these carbons from the carbon of nitrogenous bases, which are numbered simply like 1 carbon, carbon number 1, carbon number 2. So if someone is saying 1 prime carbon, you automatically will understand that the carbon being discussed in the nucleotide is carbon of the deoxyribose sugar. And another thing you have to remember is the carbon number 1 or 1 prime carbon of deoxyribose sugar, it will attach to the nitrogenous base, and this deoxyribose sugar is lacking oxygen at this 2 prime carbon. Like in the RNA on the 2 prime carbon there is OH group. Because this deoxyribose sugar is having only H at 2 prime carbon and lacking oxygen, that's why this is also called deoxyribose sugar. And this 2 prime carbon helps you to understand that the ribose sugar is of RNA or of DNA. Like if it is having at 2 prime only H, then you may understand that it is deoxyribose sugar, the component of the DNA, deoxynucleotide, and if there is OH group, then it will be present in RNA. In 3 prime carbon, this OH group will make bond phosphodiester bond with the incoming nucleotides phosphate group. And here on 5 prime carbon, it will be attached to the phosphate group. So you have to remember that 1 prime carbon is attached to nitrogenous base. 2 prime carbon help to differentiate that if this sugar will be of RNA or DNA, and 3 prime carbon, this OH group is important for the formation of phosphodiester bond, and on 5 prime carbon phosphate group will be attached. The second major component of nucleotide is nitrogenous base. These nitrogenous bases are of two types. They may be double ringed like these ones and single ringed like these ones. Double ringed are known as purines and single ringed are known as pyrimidines. In purines that there are adenine, guanine, and in pyrimidines there are cytosine, uracil, and thymine. Adenine, guanine, cytosine, and thymine are found in DNA like A, T, G, and C. These are the component of DNA, while uracil is found in RNA. And in RNA, uracil replaces thymine, and adenine, guanine, and cytosine are same in RNA also. Only in RNA, instead of thymine, there will be uracil. And how you can remember that these are double linked and these are single linked? There is a simple trick like pure as gold. Pure mean purines and as A refer to adenine and G refer to guanine. Pure as gold means purines are adenine and guanine. And for pyrimidines you can remember that but cytosine, uracil, and thymine. What we have studied till now is the nitrogenous base and the ribose sugar, and if we combine these two, it is called as a nucleoside. Like if the nitrogenous base is attached to the carbon number 1 prime carbon of the ribose sugar or deoxyribose sugar, this combination is known as nucleoside. This is nitrogenous base and this is ribose sugar. The third important component of the nucleotide is phosphate group, and in this figure you can see how phosphate group is attached to the 5 prime carbon of the deoxyribose sugar. This is deoxyribose sugar. This on 1 prime carbon there is nitrogenous base, and on the 5 prime carbon phosphate group is attached. The first phosphate is known as alpha phosphate, the second is known as beta phosphate, and the third is known as gamma phosphate. And the second and third phosphates are energy rich bonds or energy rich phosphates. Another important point is that these phosphate groups gives DNA a negative charge overall, which affects how DNA behaves inside the cell and interacts with proteins. You may have heard that histone proteins are positively charged, like suppose these are histone proteins and these are positively charged. Inside the cell, DNA in the nucleus is wrapped around the histone proteins. Suppose if this is DNA. This is DNA, it is wrapped around histone protein. How the interaction will be strong between the histone and DNA because of the opposite charges. DNA is negatively charged because of the phosphate group and histones are positively charged. So this bonding, or you can say the interaction will be very strong and very tight wrapping will be there around the histones of the DNA. In this figure you can see the complete structure of a nucleotide. In center there is the ribose sugar, and on 1 prime carbon nitrogenous base is attached, and you can understand completely this nitrogenous base is having 2 rings. That's why this will be purine. And there are 2 nitrogenous bases which are purine, mean they are having double rings, adenine and guanine, pure as gold. Adenine and guanine are purines, and this is a purine attached. There may be any base like That may be adenine, guanine, thymine, or cytosine, any of them. On 5 prime carbon of the deoxyribose sugar, there is phosphate group. We have completely understood the structure of nucleotide and its each component. Now these nucleotides will link to each other to form long stretches of DNA. Let's understand how the bond between the nucleotides is developed. In this figure you can see here is one nucleotide and on the lower side here is another nucleotide. And I am numbering the carbons of this deoxyribose sugar, like here is 3 prime carbon, and on the second nucleotide this ribose sugar is here is 5 prime carbon, and on this 5 prime carbon you can see phosphate group is attached, and on the second nucleotide on 3 prime carbon of deoxyribose sugar here hydroxyl group is attached. Now from one deoxyribose sugar, or you can say from the one nucleotide hydroxyl group will be shared, and from the other hydrogen atom will be shared to form water. And in this way these two nucleotides will be linked to each other, and this bond is known as phosphodiester bond. You may ask a question that, sir, why this linkage between two nucleotides is called as phosphodiester linkage? To understand you people, I will first illustrate that how an ester linkage looks like. Ester linkage looks like R single bond C is double bonded to oxygen, then single bonded to oxygen, and then single bonded to R. In ester linkage, carbon that is double bonded to oxygen that is linked to two R groups through an oxygen. In the case of nucleotides, here we can see a structure that there is R group, then oxygen, then phosphorus that is double bonded to oxygen, and then oxygen and then R group. And here also oxygen is there. In this figure you can see that this whole is an R group. This whole one is an R group. And then it is single bonded to oxygen, this oxygen, and there is phosphorus that is double bonded to oxygen, and then again oxygen, and then again there is a second R group. So in this bond one ester linkage is here and second ester linkage is here. That's why this bond is known as phosphodiester bond. Through phosphodiester linkage between nucleotides, long stretches of DNA are produced. And suppose this stretch is running in 5 prime to 3 prime direction, and you know that on 3 prime and on DNA there is hydroxyl group, and on 5 prime and there is phosphate group. Through the free hydroxyl group, DNA can be further elongated by adding more nucleotides and linking them to phosphodiester linkage. If I draw the nucleotide sequence here, like here is adenine, thymine, cytosine, guanine, cytosine, thymine, adenine. This is a stretch that is running in 5 prime to 3 prime direction, and if I draw another stretch of the DNA that is also running in 5 prime to 3 prime direction, and the bases are thymine, adenine, cytosine, guanine, adenine, and thymine. You know the DNA is double helix, mean they are not single strands, they are double strands, and these two strands in this configuration they are running parallel to each other, mean one is 5 prime to 3 prime direction and the second is also 5 prime to 3 prime direction. So my question is that if they have to generate a double helix through base pairing, like through forming the hydrogen bonding between their bases, like here, they cannot come in a parallel way, they have to come in anti-parallel way, like the one strand has to run in 5 prime to 3 prime direction and the second strand should run in 3 prime to 5 prime direction, so that is the way in which their bases will come close to each other to form hydrogen bonding. So I am drawing a second strand, that is this one, in opposite direction. Like I am drawing this strand here in 3' to 5 direction. Here it will be having adenine, here A pairs with thymine, C pairs with guanine, G pairs with cytosine, and A pairs with thymine, and T again with E. Here you can see that adenine always pairs with thymine, and there are two hydrogen bonds, and guanine always pairs with cytosine, and there are three hydrogen bonds. Now we have understood that how the DNA was discovered, double helical structure of the DNA was discovered, and then we studied the nucleotide and then its component like deoxyribose sugar, nitrogenous bases, and then phosphate group. We also studied the linkage between two nucleotide, how it occurs through the formation of phosphodiester bond releasing water molecule. And then now we have studied that how these stretch come close to each other to form complementary base pairing. This base pairing is known as complementary base pairing. Okay, and you know A always pairs with G and always pairs with the C. This rule was formulated by Chargaff, and the diameter of the DNA, the double helical structure, was given by Rosalind Franklin through photo 51, which Watson and Crick used, and they gave the final form of the DNA, that was double helical structure. Now we will understand the features of the double helix, and then this lecture will end. In this figure, you can see the double helical structure of the DNA in which two strands are running in anti-parallel directions. Suppose this strand is running in 5' to 3' direction, then this strand will be running in 3' to 5' direction. In this way, these strands twist upon each other along the whole length of the double helix, and they form certain grooves, wider groove and narrower groove. Wider grooves are known as major grooves, and narrower grooves are known as minor grooves. And if we start from the here, then this length of the DNA till here, it forms a complete turn of the helix. If I draw it separately, then it will be like this. If I start from here, it will be a complete turn of the helix, and its length is 3.4 nanometer. In angstrom, it is 34 angstrom. But the distance between the stacked bases is distance between the stacked bases, and it is 0.34 nanometer. And also you have to remember the diameter of the DNA, that is 2 nanometer. And this diameter is uniform. What it is? It is uniform. Why it is uniform? Because if on one side there is a single linked base, in the complementary side there will be double linked base. Like if there is cytosine, there will be guanine, that is double linked. If there is you can say adenine, that is also double linked base. If there is adenine, there will be thymine, that is single linked base. So in this way, the diameter of the DNA along the whole length remains constant, or you can say uniform. This complete turn of the helix is also known as pitch of the DNA, or pitch of the helix. Another thing you have to remember is the backbone of the DNA. This is the backbone of the DNA. I can highlight it. This is the backbone of the DNA, and it is made up of sugar plus phosphate. Sugar plus phosphate. DNA backbone is made up of sugar plus phosphate. Sugar is deoxyribose sugar and phosphate. But the nitrogenous bases are embedded between the backbones. These are nitrogenous bases which are embedded in between these backbones. And from this double helical structure, the model of DNA replication that was semi-conservative model of DNA replication that was quoted by this structure. Like when DNA structure was discovered, then many scientists gave their models that how DNA will replicate. Some of them gave the semi-conservative model of DNA replication. And the structure, which was double helical structure, supported this model. In this way, the structure of the DNA was discovered in a step-by-step discoveries and the contribution of the science. Thanks a lot for watching, like and subscribe.